Hello students, I am Dr.A.Basker, Associate Professor, Chemistry from KIT, Kalaignar Karunanidhi Institute of Technology. In this video, we are going to discuss about glycogenesis process. What is mean by glycogenesis process? It is otherwise called as glycogen biosynthesis. When the excess amount of glucose produced in our body automatically converted into glycogen. Glucose is a monosaccharide molecule. It combined to form a polysaccharide glycogen. This process occurs in all the tissues in our body, but the major portion takes place in liver and muscles, and a small amount of glycogen also synthesized in the kidney. It is one of the very essential process for the energy requirement. When the body produces excess glucose, automatically it will be converted into glycogen. At the time of energy requirement, the glycogen molecule break down to convert it into glucose. So the conversion of glucose to glycogen is called glycogenesis. In other words, the glycogen is converted into glucose is called glycogenolysis, otherwise called as glycogen breakdown. How the glycogenesis process, the glycogens are produced from the glucose. So actually 5 different steps are involved in the reaction. In the first step, glucose is converted into glucose 6-phosphate using the enzyme called glucokinase and also hexokinase. Glucokinase is the enzyme present in the liver and hexokinase enzyme present in the muscle. So either glucokinase or hexokinase reacted with the glucose molecule, the ATP is transferred one of the phosphate group to the glucose molecule and ATP is converted into ADP. ATP is called adenosine triphosphate, ADP is called adenosine diphosphate. So after the releasing of phosphate group to the glucose molecule, the ATP is converted into ADP. So glucose molecule is a 6-membered ring system. So one of the hydroxyl group from the glucose molecule is replaced by phosphate group. So in this reaction, phosphorylation of glucose takes place, leads to the conversion of glucose 6-phosphate. After the conversion of glucose 6-phosphate, the molecule undergo mutation by using the enzyme called phosphoglucomutase. Mutation in the sense glucose having the phosphate group in the 6th position, automatically it is changed into 1st position. So both the glucose 6-phosphate and glucose 1-phosphate are equilibrium to each other because of only the positional change. So same the glucose 6-phosphate and glucose 1-phosphate both are having the same molecular formula, but only the change is positional change. And also the activator, magnesium ions and coenzymes glucose 1,6-phosphate also involved in the reaction. So here phosphoglucomutase is a protein and glucose 1,6-diphosphate is a non-protein part. You know that phosphoglucomutase is a enzyme and glucose 1,6-phosphate is a coenzyme involved in the reaction. It is a supporter of the enzyme for this reaction. So in the step 3, glucose 1-phosphate is activated and it is converted into uridine diphosphate glucose. Simply it is called UDP glucose. So where we obtain this UDP glucose? From the UTP molecule. UTP molecule releases UDP and pyrophosphate. The released UDP molecule attached to the first position in the glucose molecule. So instead of phosphate group, it is converted into UDP group. So the glucose 1-phosphate is converted into UDP glucose. This reaction is nothing but a hydrolysis reaction. Hydro in the sense water, lysis in the sense breaking. So when the hydrolysis takes place, the UTP molecule produces UDP and pyrophosphate. So after the formation of uridine diphosphate glucose, it starts forming glycogen chain. So forming the glycogen chain, the UDP glucose molecule releases UDP from that same molecule and start forming the glycogen chain. So the maximum unit level present in the glycogen chain is up to 8 unit. For every chain in the glycogen molecule, we have 8 same glucose molecule. So the 9th molecule not paired the 8th molecule because it start forming another chain. For one chain we have 8 glucose molecule. In the 9th molecule start forming another chain. The two chains are interlinked by 1,6 linkage, glycosidic linkage. And between the two glucose molecule, it is linked by 1,4 glycosidic bond. So after start forming the first chain, the second chain is linked with 1,6 linkage. Within the chain, that is intra-chain linking is 1,4. Inter-chain linked is 1,6. So after forming the chain, the glycogen is produced from the glucose molecule. Similarly, in the glycogenolysis, the glycogen molecule break down and produce the glucose. The produced glycogen molecules automatically stored in livers and also muscles. Thank you for watching this video.